I'll be teaching you today about the central dogma of biology. Now, the central dogma of biology has two major processes called transcription and translation. Now, the importance of these processes is that they are the ones by which the genetic code is collecting enzymes in order to produce all of the proteins in an organism. The transcription happens in the nucleus, while the translation will happen in the cytoplasm. To start our diagram, I will split up the nucleus and the cytoplasm by the wall. And then I will go over to the nucleus. In the nucleus, we have 46 chromosomes, 23 pairs, half coming from the mother and half coming from the father. Now, inside of our chromosomes, we have our genes, where the proteins are produced. And our proteins then will be produced throughout our DNA, which is also our genetic code. Now, the first step in producing proteins is that we have to have our DNA synthesize our RNA out of it. And our RNA gets synthesized with the help of an RNA polymers, which will then unwind the DNA going to the right. So once this starts happening, it starts spitting out the synthesized RNA. But this is not yet complete RNA. There's actually three different types, which are our amino acids: RNA, tRNA, and rRNA. So the mRNA then turns into our pre-mRNA after it gets out of the DNA. From here, we are not quite done, and we do not have our full mRNA yet. First, it has to undergo the process of splicing, which is removing inputs. Once the splicing has occurred, we finally have our mRNA, which will then leave the nucleus and then go out into the cytoplasm. Now, we're going to pause there before continuing on and come back to our nucleus, where we will form our tRNA. Our tRNA, again, is synthesized out of our DNA and then made into the pre-tRNA, which gets spliced and then has our final tRNA, which then will leave the nucleus as well into the cytoplasm. Now, back to the mRNA. It comes out into the cytoplasm, waiting for it to be used as the template for further protein production, while the tRNA comes out in a weird bunched form. Now, the tRNA will then attach itself to a single amino acid, of which there are 24 different types inside of the cytoplasm. From here, we have our two templates for making our new protein, but they cannot be combined on their own. To do that, they need to be combined with the ribosome. A ribosome is made out of nucleotides in the nucleus as well, and then it moves itself out into the cytoplasm and attaches to the cap of the mRNA. Now, the mRNA is positioned from the 5 to the 3, and the ribosome will continue down that pathway, attaching random tRNAs into the mRNA, matching it up with its opposite letter. For example, the T would attach to the A, or the C would attach to the D. And then once it starts moving down, we have our half of our genetic code. And then it needs to go back down the other way, from the 3 to the 5, even though it cannot completely go that way. So we have to have it flipped upside down, so it's still going from the 5 to the 3, where it will be filling in the missed spots from the ribosome connecting the tRNA to the mRNA. Now, after this process is done, we'll have some leftover proteins which will come out of the back end of the ribosome and then get rebroken down into more amino acids and attached to other tRNAs, which will then restart the process and then go over into making more of proteins. So the proteins are important because we need them to make more DNA.